Afternoon, everybody. So I am Dr. Prasoon Kumar, Assistant Professor in the Department of Biotechnology and Medical Engineering at NIT Rourkela. And I am going to present one of the work which was translated into a product. And this work was done by my student Vaibhav Shitole. So the title of the project is Microporous Volumetric Sampler to enable sampling and storage of blood for dried state for pharmaceutical analysis. So I own a lab where biodesign and the medical device laboratory, where my research area is biomimetic design, manufacturing technique, and the polymeric materials. So I work on the majorly medical devices. So coming to the presentation, in the today's talk, I would be discussing about the background story behind this innovation, then the problem definition and the motivation behind it, how we ideated into the idea, and then what's the experimental plan and the results of the, and finally conclusion and the future forward. So looking at the background story, a few years back in 2018, a lady named Dr. Vaibhav Sridhar, she is the founder of Molecular Solutions Care Health. She came to the IISc Bangalore where I was doing my postdoc, and she said that she used to do molecular diagnostic tests, and for that she needed the blood samples. Now, if the blood sample need to be transported through the cold chain transport, and it requires 3 to 5 ml of the blood withdrawn with the help of the venipuncture and specialist, typically it requires the transport issue like the cost, and the larger volume is the blood is required. Sometime during the emergency, she could not get the blood. So she came up with this problem to me, to how we can address this problem. Now she had one of the solution which is currently available in the market, that is called the dried blood spot. But in this, there are the number of problems are there, like a single drop of blood is required, homogeneity is not there, not multiple touches, then only you can get the perfect spot. And it takes about 4 hours to get dried in a humid conditions. So my sample may get degraded. So the problem associated with the current dried blood spot is the risk of contamination, uneven coverage of blood on a paper card, need a trained professional to do it. It's a time consuming as well as the risk of the biohazard. To solve this issue and to understand the magnitude of the problem, I found that the biological specimen collection is a very big issue in diagnostic industries. Moreover, it is also required in the clinical trials, pharmacokinetics, pharmacodynamic profiling studies, as well as the basic research. And the blood is one of the most important biological specimen for doing this analysis. So if you look at the market size, blood sample collection and testing itself is growing at a huge rate, and there is huge need of the blood collection in number of tests like the molecular diagnostic and in the clinical research. And we cannot afford to get for the cold chain transport, which is costly, time consuming. So we thought of where it could be used in the pharmaceutical as well, as I was there in the Naiper Ahmedabad at that point of time. So in the pharma industry also, there is an issue of the blood collection and the testing. And primarily it's used for the drug discovery and the development process. So drug toxicology studies, then the therapeutic drug monitoring, preclinical and the clinical practices do require these type of the blood. And in the pharma industry, they are also using for the preclinical and the clinical. If you look at retroorbital way of blood drawing, it requires a painful collection, need trained personnel. Moreover, taking a blood from a pediatric and geriatric patients is also a big challenge. So we need, nowadays, analytical techniques have been too sophisticated that we require only a drop of blood. So taking out 3 to 5 ml of blood from a pediatric and geriatric patients is too uncomfortable. So we hypothesized that if we can come up with a device, a 3D hydrophilic porous material with defined pore size, it can automatically suck a drop of blood through a prick of a finger, and we can dry it and ship it to the different laboratories for the testing and analysis. So with the help of this MSc student who worked with me, we developed this device. So how we did it? So we created a specialized hydrophilic polymer material with the want of IP protection. I am not going to share the chemical name. but we had some hydrophilic polymer material, and that we tried to polymerize to form a microporous structure, which you can see through the same images. And these microporous structure were tailored and tuned so that the microporosity is consistent. Then we tried to understand how, if a blood is being drawn, how well it can suck the blood, irrespective of the hematocrit volume. So if the hematocrit volume is going to change, then the sucking of the blood by the porous matrix by the capillary action would be poor. So when we checked with the different hematocrit from the 30 to 70 hematocrit bias, we found that it's a non-significant, and our matrix was able to suck the blood in a better way. Then we also tried to understand that if a person has to self-sample, how well it is variability between the person to person sampling. So we took 5 test person who could draw the blood with the heart of our device, and we found that microliter of the blood absorbed didn't get changed with the different operators using our device. Further, we tried to dry our device in the different conditions, like controlled closed container with the help of a desiccator and the control open area. And we see that in the case of the desiccant, our device would provide 100% within the 4 and 5 hours, whereas the control and the control open air took longer hours to dry. And this is very much essential if the sample need to be collected from the hot and the humid condition where sample may get degraded. It should be dried quickly to be easily transported. Then we check for the material fabrication consistency. For the scalable production, we need the matrix to be produced with good consistency, like the porosity is the one important factor. And we found that in the 5 batches we tested, the porosity was quite consistent with whatever the parameter which we have optimized. So then we converted this matrix into a usable device using the CAD design and the 3D printing. So you can see here the, so you can see on the right hand side, there is 3D printer microsampler device. So now the question was that if the blood is being automatically stored in this blood microsampler, can we do the analysis? So there we have Dr. Pinaki Sengupta. He is a professor in NIPER Ahmedabad, expert in the pharmaceutical analysis. With the help of him, we conducted this bioanalytical test on our device. So we spiked the blood with the caffeine as a drug molecule, then sampled it with the help of our blood microsampler, and then stored it in ambient temperature, then extracted the caffeine drug with the help of the solvent, and then did some analytical tests. So first analytical test we did that we wanted to extract the blood with the help of column chromatography, and we were able to find at what in the graph you can see the chromatogram of the caffeine, which is being seen as the 6.7 C, and this is consistent for all our samples. So we understood that at this elution timing we are going to get our caffeine. So further we checked the linearity of the samples for the different type of the samples, and we found that for the varying concentration for 2 to 12 different concentration range, the sample was quite extraction procedure was quite linear in nature. Then we checked for the stability of the analyte, because the analyte need to be stored and transported and then it to be extracted. So how far it could be stored. So from the fresh sample to sample collected at the 15 days, we were able to see that the stability was quite good. And finally, the extraction recovery. So after stored for a long period of the time, can we extract the amount of volume? So when the concentration is quite high, like 10,000 nanogram per ml, we were getting our 95% recovery, where 600 nanogram per ml, we were getting only 71%. This is data is good enough to start with. So in conclusion, we have developed a unique material which can draw the blood and store it and in a dried format. We characterized our device, did some quantitative bioanalysis, like the sensitivity, extraction, recovery, and the stability of the drug. And for, yeah, 1 minute. So future is that we are exploring this microsampler device for the different type of the blood sample collection and uses, like sputum, saliva, tear, sweat, semen. And we are aiming to use this device for the molecular diagnostic, therapeutic drug monitoring, illicit drug monitoring, and the newborn screening. So these are the couple of awards and recognitions which I have got for our device. And recently this product was being launched in the December for as a marketed product based on this innovation. So apart from this, I work on the number of different innovations. And thank you everybody for your patient listening.